I'm a PhD student at the Center for Integrative Ecology, and I work with Matthew Simons. What I want to see is whether related species tend to produce similar sort of chemical compounds. In other words, whether related species can be medicinal based on their chemical similarity. So my study group especially for this study is the magnolids, which is an ancient group, and from that group about, say, in Australasia alone, about 20% is about medicinal species, which are used in indigenous medicine. So which makes it an interesting group to study in terms to see whether evolutionarily related species tend to produce similar sort of chemical compounds. Well, I chose this research because I'm very interested in native plants, and most of these magnolids in Australasia are native to our country. Whereas if you consider my early background, where I come from, where I've also been studying native medicinal species, which got me very interested in knowing how these medicinal traits have developed in plant species. So if we consider in today's world, there is a rapid decline in the use of plants in order to develop lead compounds for pharmaceutical industry, because in the world there are about 300,000 plant species, and we don't know where to look in terms to find a medicinal candidate. So in that term, if we can narrow down this process of looking for a medicinal plant species, we would be able to find a lot of pharmaceutically potential compounds through this plant kingdom. So that's something interesting in this research work that I'm actually doing. In this project, firstly, I needed to identify species. In Australia, there are close to 350 magnolid species. So it is hard to sample all these species. So my initial step was to build a model to pick a potential species for my analysis. So I narrowed down it to half of the clade, that means 150 target species, and I wanted these species to come from a uniform environment. Therefore, the first step I developed was collaborate with botanic gardens all over Australia and New Zealand in order to see whether these species from this magnolid group, whether they have them in their collections. So that was the very initial step, and it was very interesting because I got to collaborate with a lot of people from botanical gardens and so on. Then, after collecting all these species, what I did was I did a big chemical analysis in order to extract all the chemical compounds to these 150 species produced. Then I did a phylogenetic analysis where I extracted the DNA in order to construct a phylogeny to see the evolutionary history. And finally, what I want to apply those chemical information into the phylogeny in order to see whether the related taxa produce similar chemical compounds. As an example, say you have X, Y, Z species, and if they produce similar chemical compounds in the phylogeny, you'd be able to say, okay, now X is similar to Y in terms of their medicinal properties. What I liked most when conducting the research was the collaborations I made with the botanic gardens and the sampling process, actually going somewhere, picking up the species from their environment, identifying those species. And then when I brought them back to the lab, I did an extensive literature search on the uses of these species. And I looked back into the literature where the traditional indigenous people, like the Maori or the Aboriginal people, have used these as their medicines. So it was very fascinating, and it was very interesting to me to learn all these uses of species that are just lying there and nobody really cares about them. So that's something very interesting that I found in my research work. When I got all these species together, the hardest thing was doing the chemical analysis for all these 150 species, because it was the magnitude of the data set was so high. Analyzing this data in a computer system is the hardest thing. You have to narrow down this data in order to make sense out of whatever the data that you have got. So that is like the hardest step. After collecting these species, getting them into the lab, then even the lab work is also interesting. But when you are going into analysis, you have to make sense of the whole data set. So I reckon that was the hardest part. So what I found is, in terms of the chemical similarity, I found that distinctly two families, that's Atherospermataceae and family Lauraceae, tend to cluster together in terms of their chemical similarity, which means that species in the family Atherospermataceae are chemically similar to each other more than it is chemically distinctive with the Lauraceae. These are the plants group where you have the Atherosperma species, which is very common in Australia, and even the bay leaves, and you have cinnamon. So that's one of the major results that I've found from my preliminary studies. But I have another 8 families which I need to analyze into and see whether they also tend to cluster in terms of the chemical similarity, and also then I need to map it onto the phylogeny in order to see whether their evolutionary history also correlates with their chemical similarity. In terms of conservation, as I mentioned earlier, there are about 350 species of this whole magnolid group, which are very ancient species. But out of these 350, only about 20 that we today speak about more generally. So there's another, say, 250 species which we are not interested about, or like we are not informed about. So this kind of research, I think, brings out the importance of these species. Say, if I am able to define a particular understudied species as being medicinally very important, it produces a chemical that is used to treat, say, cancer. So then the species is brought to limelight, and then you have the mindset of conservationists is toward the species in order to identify it in the wild and to see the conservation status, and it sort of highlights the importance of the clade in terms of being medicinal. Then it again then it again also highlights the need for a conservation strategy. As an example, like today I heard that the northern white rhino has been extinct. So it has been poached since 1970s, and it was widely discussed only during the late 1990s because of the importance of the species. This is because the species was so outnumbered in the wild. So before we move there, this kind of research highlight the importance of the clade for us to look at it in the wild and see what is the conservation status. Where should we take this species? It takes the species to a next level, I think. So in that terms, it's important in terms of conservation as well. And moving on to the pharmaceutical industry, this is where it's most important, I think, because today we don't have a particular method of picking out plant species in order to trace them for pharmaceutically important chemical compounds. But if we are able to relate these medicinal properties of plants into their phylogeny, we have a particular method of picking out a particular plant species which should be highly medicinally important in contrast to other taxa and plant families of the plant kingdom. So in terms of that, in order to find more pharmaceutically important lead compounds from the kingdom Plantae, this sort of research puts a big stepping stone, I guess. Currently, there is no global list of medicinal plants. So what I suggest is, if we can combine all the knowledge from the traditional and indigenous communities, like you have Chinese medicine, you have Ayurveda, and medicinal knowledge of Australia, America. If we can combine all that knowledge and make a checklist of all these medicinal plants and what they are used for, and again then there are like research that's been done in the past decades on the active compounds of these medicinal plants. So if we can bring all this information together and make it publicly available on a particular database, I think that would make more sense in making it available for the public, and also it makes it easier for the researchers as well to pick up species in order to investigate further in terms of developing pharmaceutical candidates and drugs and so on. After this research work, I think it would be a stepping stone to like lead discovery in pharmaceutical industry. So I don't like the concept of research just lying there. I want it to be used. I want the research to be useful for the future generations. So one thing I want to do is in terms of this research work which I've been doing, I want it to be publicly available, and what I'm trying to do is I want to collaborate with the botanic gardens and make this information available. And there they have particular there in the botanic gardens they have public information leaflets that they distribute when people come to this botanic garden. So what I've suggested this botanic garden is, if I find that particular species that I've collected from their gardens are useful, I want that information that I've discovered to be publicly accessible and available at the botanic garden, so that that people know what the plant that they're looking at is useful. It has some sort of a medicinal history. It has a medicinal benefit for the humankind. So that's one thing in terms of my research and in terms of the future of my research. And if career-wise, if you're talking about me as a researcher, what I would want to do is continue into this native plant research more. So one of my main interests is looking at these medicinal plants from its native environment, from Australian forests and so on, and collecting them and looking at their chemical diversity in terms of their environmental impact. So my main, because I like research, my main aim for the future is like continuing in the same path and studying the Australasian native plants.